I'm Nancy from the County Library, the South Jordan branch, and I'm here again today with Dr. Philip Moos, and today he's going to tell us about mutations.
Some part of the research that I'm involved with is part of the Cancer Systems Biology Consortium, and what we're interested in there is what we call genomics. And this is actually trying to look at what are all the changes that occur in DNA and RNA when you have a situation like cancer develop.
Okay, so last time you told us about how we get from DNA to proteins and transcription and translation. So for mutations, where in this process does that happen?
So that's an excellent question. And if you remember from last time, I was using these 3D printed manipulatives from EagleKnot from Thingiverse, and I'm going to use them again in this context. So as we had last time, we had a strand of DNA, and I just have one strand this time. And so if this is our strand of DNA, so this would be the complementary strand of RNA that would go with that, right? So if we had a substitution of one base, that would cause the RNA to be generated differently, right? So because we still need that A and T to match. So this would be a mutation at in one base.
And that's called a substitution?
So that would be a single base pair substitution, yes. So remember this, before this had a U here, so this would match this codon, right? But now with an A here, it would now match a different codon. So this could result in a different amino acid getting inserted into the spot, right? So we can have insertions or deletions as other means of creating mutations. And so this then would change how the RNA is being interpreted, because we have this codon and then now a completely different codon for a different amino acid following that one.
So how many amino acids are there?
So we like to think about there being 20 standard amino acids. And if you're really good at math, you might be able to tell that that these codes of using 3 bases, that there's more possibilities than 20 possibilities for that, right? And what we find is that some amino acids are coded for by up to 6 different codons. Some of them are only coded for by 1. So this can very much change the properties of the protein that's being made off of this by changing what the amino acid sequence is. Okay, the last mutation that I wanted to talk about was a deletion. And this then makes so that now we only have one amino acid. In fact, this isn't actually an amino acid. This is a stop codon. So this would actually terminate the protein from being made.
So now you've shown us about these different types of mutations. So that makes me think, how similar are people's DNA?
It's an excellent question. Humans have about 99.9% identical DNA.
Wow.
The little bit of variation that we see, so you get different slight variations in their sequences, create some of that, and then you have disease situations where these mutations can accumulate. Cancer is a perfect example of that, because cancer accumulates many, many mutations, and that's what we study when we're doing systems biology.
Wow. Well, that's really interesting. Thank you, Dr. Moos. If you want more information, we have some excellent resources on our website at thecountylibrary.org. Thanks for joining us.